Good evening. My research project is the analysis of genes related to the progression of Huntington's disease. Huntington's disease is a dominant neurodegenerative disease with typical clinical manifestations of choreographic movements, mental disorders, and dementia. In general, the onset of the disease is in middle age and will eventually lead to death after 10 to 20 years. Right now, doctors can only diagnose HD by early screening and delay its progression, but there is no effective treatments. In my project, I compare and analyze the blood samples of different stages of the Huntington's disease patients and normal people in the GO database and screen the genes that are different from the progress of Huntington's disease. The blood sample chips were retrieved and analyzed from the GO public database, and gene differences were analyzed by software tools such as GOR2, GO, KEWG pathway, and PPI network. The key differential genes of HTT gene were analyzed to obtain the information of the relationship between microRNA and the target genes. Then the differential expression of microRNA and mRNA was analyzed to identify the key microRNA gene. The results were displayed through the microRNA target gene network regulatory map through the integration of the two data. The regulatory mechanisms involved in the expression of the process of HTT key genes were explored. In my research, the upregulation of 5 target genes is related to the progression of HTT, and we can focus on the clinical analysis of these 5 HTT genes and the development of gene intervention therapy drugs.